Hi, I'm Morgan, and thank you for coming to updates on my dissertation work studying how bird microbiomes relate to health. So, we all came here for some animal behavior, though, right? Why the heck is this guy about to talk to us about bacteria? Well, it turns out behavior can be influenced by the bacteria in an animal's gut microbiome, which is a community of naturally occurring bacteria living in our intestinal systems that every animal develops. Now, we call this connection the microbiota-gut-brain axis. However, if they never develop this normal bacterial community, animals can develop a whole suite of abnormal behaviors. For example, germ-free rodents raised sterilely in a lab have all sorts of behavioral and neurological issues, but lactobacillus probiotics have been shown to wipe away these deficits if administered before a certain developmental age. Now, for the first part of my dissertation, I tested for this connection in songbirds and found that zebrafinches' performance on learning and memory tests differed depending on their microbiome structure. This got us thinking about the underlying physiology that might be at play, so we expanded our research to a more holistic perspective. So, thinking more broadly, in addition to this direct communication along the gut-brain axis via the vagus nerve, the gut microbiome can affect the brain in indirect ways too, such as from a stress response mediated by the hypothalamic-pituitary-adrenal axis, or HPA axis for short. The stress response is evolutionarily a good thing. We've evolved it, to save us from the things that caused us stress in the first place. But a harmful side effect is that the release of glucocorticoids, or CORT, triggers an immune response that can indiscriminately wipe out the body's microbiomes, which opens the door for opportunistic pathogens and may alter communication with the brain. This connection between the gut microbiome, cognitive function, and stress has spawned the framework the microbiota-gut-brain HPA axis, with the health of the host lying in its center. And gosh, is that a mouthful. Which makes sense because there are so many systems involved here. It can be really tough to tease apart, how does the HPA axis affect the brain and vice versa? What is the directionality of these arrows? How does all of this come back to reflect in the health of an organism? That's what this dissertation is aimed at teasing apart, is the directionality of these arrows and how one system relates to another system. Ultimately, how can we observe this in the health of a living organism? So we've reached our central question here. Can you change an animal's gut microbiome by inducing a stress response? We also wanted to know what other aspects of health are affected. And most importantly, can you identify any consistent microbiome differences between stressed and healthy individuals so that one day you can improve animal health by resolving these microbiome differences? Now, because of this ultimate goal, we wanted to get out of the lab and study wild microbiomes, so we chose the Northern Cardinal. To answer these questions, I'm following a repeated samples within individuals design, where I capture a cardinal to take my samples, then release it, and then induce stress, and then recapture and resample it. Male Northern Cardinals make a good study system for this question because they're highly territorial and will respond aggressively to territorial intrusions, which we can simulate by playing recordings of male cardinal song to lure them into a net to sample and tag them. This capture event also reliably produces an acute stress response, which we can study from glucocorticoid hormone concentrations in blood samples taken during capture. Now, the graph here shows the Northern Cardinals' increase in CORT as a result of capture and handling. Their bright red-orange beak is also a carotenoid pigment-based sexual signal, whose coloration is related to individual quality and will actually fluctuate depending on the bird's current physiological state, such as after a stress response. I'm doing this work in Treetops Park in Davie, Florida, where the cardinals are practically dripping from the trees. I'm predominantly working with males because they're reliably territorial, and I color band each male that I catch. I take a series of blood samples immediately after capture for a CORT baseline, and again at 15 and 30 minutes after the first sample, so I can estimate the slope of the CORT response. I take standardized photographs of the beak to assess coloration in ImageJ, and then I take a cloacal swab, which they're not a fan of, but then an oral swab, which they get to bite, and that's every cardinal's favorite, so they do like that, followed by the standard battery, of course, of body measurements. When I release each bird, it will be assigned to either the treatment group or the control group. Now, control birds will be left alone for 1 to 2 weeks and then recaptured to resample everything. And treatment birds will receive the social stress treatment, where I set up a male song playback for 30 minutes every other day, and then they're recaptured for the follow-up samples. Because so little is known about how bird microbiomes relate to the samples that I'm taking, and because my field site was shut down for COVID, which was so much fun, and then reopened as a vaccination site, which was even more fun, I swabbed basically any cardinal I could get my hands on, including males, females, fledglings, nestlings, anything. This will help me characterize the core microbiome of the Northern Cardinal and how it differs between sexes and age groups, and also observe its baseline relationships between court, coloration, and body condition. This has given me a very crowded -80 freezer, and unfortunately microbiome sequencing is an absolute monster of a process, and supplies are backordered, so I won't have any sequencing results for you today or for another few months. But stay tuned. I want to spend my last few minutes reviewing what we expect to come from this freezer full of samples, and to really drive home the potential good that this research can bring to wildlife. So we have our first 3 health metrics here, the beak coloration quality, body condition, and court, and we expect to see a deficit in each of them after that first capture, regardless of treatment group. For example, beak coloration should decrease, as should body condition, and court should increase. But you'll notice some differences here between treatment and control. If our treatment was effective, treatment birds will show sharper deficits than controls because of that added social stress from our playback. But moving on to the microbiome data, first we have alpha diversity, which is the number of different types of bacteria within a single microbiome sample, and it has been shown to correlate positively with host health. So high alpha diversity is good. Diversity is expected to decrease with the stress of capture for treatment and control birds, but as with the health proxies, we expect this dip will be greater for treatment birds compared to control birds. Beta diversity gets more complicated and is often more important because it measures diversity between samples, not within samples. So you can observe how microbiomes cluster by similarity within each sample group. It's based on taxonomic relatedness and relative abundances, so we can detect if certain sample groups have abnormally high abundances of, for example, pathogens. Starting out, we expected the initial samples for control and treatment groups will be very similar because we haven't done anything to them yet. This is the natural cardinal microbiome. But we predict that community structure will shift between the first and second capture, and that treatment bird samples will cluster away from those of control bird samples. Ultimately, this project is dedicated to improving wild animal health. Now, one of the most common reasons that a patient dies in a wildlife hospital, aside from its injuries, is because it's simply too stressed to survive the care that it receives, because it must remain in captivity for its care. If we can figure out what specific bacterial species influence the differences between healthy and stressed birds, maybe we can design probiotics to remedy this shift. Unfortunately, we're years away from a working real-world application of the problem, but this is the first step that we need to take so that we can learn the underlying mechanisms in which the microbiomes mediates animal health. Thank you for your time during this busy virtual conference, and a big thank you to the many funding agencies that have helped me chip away at the high cost of hormone and microbiome analysis. This includes the Animal Behavior Society, so thank you very much. My email is at the top of the slide here if you have any questions.